Now that we've talked about the comparison between RNA and DNA, let's look at how DNA replicates. We know that DNA is a double helix that's bound by weak hydrogen bonds. And we're going to take some advantages of those weak bonds and we're going to unzip it and use each strand as a template to make another strand. So if we watch this animation, we can see that we have a nucleus, we have chromosomes here, and DNA replication is the process that a cell uses to copy those chromosomes before it divides. And that's back, when we talked about mitosis. And all the chromosomes, remember those chromosomes are DNA that has been condensed. So the cell's going to make a copy of those so that each cell can have new DNA, and that's where this process comes into play. So we're going to just select a segment of DNA to do this with so we can see how it's replicated. And we're just going to look at a really small piece of it because there's so much. And if we zoom in on this piece to see how it replicates, we see that we have our double helix, the two strands. We have the nitrogen bases here, and we have our phosphate and sugar deoxyribose backbone here. And that goes all the way down, and that's duplicated because we have one on each side. Now, what happens is the DNA is going to unzip to form two single strands, but then it doesn't completely unzip. The process is ongoing. So it partially unzips, and we start the replication process as it's going through so it can re-zip up. So we have our two strands, and each contains the information to make that duplicate copy. And remember, they're antiparallel. The enzyme that actually is going to break the hydrogen bonds apart to split these strands is helicase. So think of helicase, remember case is an enzyme, the ASE, and think of helicase as the scissors that are going to cut the DNA. Now, subunits of the DNA are going to be attached to the old chromosome, and so we're going to end up with two double strands. And then we're going to attach these new subunits. And remember, we take in a lot of these nutrients, and our cells are going to break them down so that we have the needed materials to do this. So these new subunits are going to match up with the complementary bases. So we know that A goes with T and C with G. Now we get an exact copy twice from the original that we had. And there's another enzyme that's going to add these nucleotides or the bases to each other, and that's going to be the polymerase. So it's like the helping hand. So if we shrink these back down to see what's happening in the whole cell, now we have two pieces of DNA where we originally had one. So we can see that all the chromosomes were replicated, and then the cell's going to go into either mitosis or meiosis. Now, if we go back to our diagram here, this is what I was talking about with the partial unzipping. If you look at this section here, that part is unzipped, okay, the helicase unzipped it, and this part of the strand is still together. The polymerase is going to be that helping hand to get those new subunits in. Remember that A is going to pair with T. If you remember at Grand Central, AT goes together, and Grand Central, the G and the C go together. Now, we can look at another diagram to build the DNA molecule, and this one is a little easier to look at and just shows you the base pairing. So if we look at the side view, we see now we're seeing the top view, and you can just go and drag these if you go to this website and practice. So T, you'd see would match with A, that's its complement, and then A with T, C with G, then again A with T. And this process keeps going throughout. And if we were to keep going at this rate, it would actually take all day and all night in order just to replicate the size of the strand that's on the side of your computer screen. So we're going to stop there. Now, let's take some notes. If you were to look at this, again, we'll review. Think of scissors, helicase. Okay, remember the ACE is the enzyme um, that's going to cut the two strands of DNA apart. Okay, now our helping hand that's going to help place the nucleotides to each of those strands is going to be polymerase. Now remember, again, you have ACE, so it's an enzyme, and that's going to add the nucleotides. And remember, if you don't remember what a nucleotide is, that's what this structure is here. It consists of a 5-carbon sugar, a phosphate, and a nitrogenous base. And this is going to happen by the law of base pairing, which states that only certain bases are going to pair. So A with T and C with G. And then the last one, the glue that holds all this together is ligase. And again, ACE is an enzyme. It's going to seal the DNA back together. So you have that double strand, again, attached by those hydrogen bonds. So we end up with 2 new strands. And we say this is a semi-conservative method because it's going to maintain the integrity of the original strand, and you're not destroying anything because we can't destroy matter, but you're going to still maintain part of that strand, and then you're just going to build onto it and make a new strand.